Okay, so this is chapter 14, and in this chapter we're going to be talking about the structure and function of DNA. And in this first part, we're going to focus our attention really on how it was figured out that DNA is the molecule of heredity. So before we jump into that, I just want to talk about some practical applications of DNA, and you've probably heard of some of these. So, modern day advances in DNA technology started in the 1980s-ish. DNA was discovered as the molecule of heredity in the late 1800s, and then the structure of DNA was figured out in the 1950s. And again, around the 1980s is when we started to, make some really big advances in DNA technology. In 1996, you may remember if you were alive, I remember pretty vividly, the first large mammal was cloned, and it was a sheep named Dolly. And Dolly actually didn't live very long. I think Dolly died in 2003-ish due to problems with the sheep's lungs. However, again, it was the first large mammal to be cloned, and Dolly actually went on to also have offspring of her own. So it's pretty remarkable. To date, we haven't cloned a human, and there's, regulations around that. There was actually some claims in other countries of cloning of humans, but those claims were not found to be valid. We clone human cells all the time inside of, petri dishes or test tubes, but to clone a whole human, that has not been accomplished yet. However, since 1996, we've cloned a lot of other mammals. So, cats, I think horses. There's a whole slew of different mammals that have been cloned. There were many failed attempts, prior to Dolly being cloned. It took, years and years of research, and again, many, many tries before it was successful. But we've been able to do it, which is pretty cool. Some more practical applications, aside from cloning, what else have we, been able to do since, the structure of DNA was figured out? In more recent years, we've been able to do gene editing. You may have heard of CRISPR technology. And in fact, just not that long ago, the first disease was pretty much, cured, as far as we know, by using CRISPR technology. It was used to actually fix the genes that cause sickle cell anemia. And so that's being used. Some other practical applications that you've probably heard of are in forensics. And so, one of the big forensics cases that I can remember is the O.J. Simpson trial. And forensics, were done to try to figure out who was at the crime scene. And there's a lot of things that happened in that case. But, we use forensics. We use DNA technology in forensics, so it's applicable there. We also use DNA technology when we do paternity testing. So we are comparing, genetic information from an offspring, with a potential parent. That comparison gives us, the answer to whether or not someone is the father. We also use DNA technology when we do genealogy. And so some of you guys may have done like 23andMe or one of those other companies where you send, a cheek swab or a follicle of hair. And that's going to contain your DNA. They can take that DNA and put it into a database with other people that have also submitted their DNA, and you can tell kind of who you're related to and what, country of, origin your DNA has. And so that's fun. We've also used DNA technology in vaccine development. So, with the latest COVID vaccine that was developed, you heard that it was an RNA vaccine, an mRNA vaccine. So again, genetic studies have led to that. Another practical application of DNA technology is disease susceptibility. And so now, if you have a family history of breast cancer, for example, you can be screened for the actual gene that increases your susceptibility for that type of cancer. And so again, just a short list, really, of practical applications of DNA technology. And so now we're going to get into some history. And let me move myself. Over here on the left is this beautiful picture. It's an electron micrograph actually showing DNA replication. And so this structure right here is called a replication fork. And we'll talk about DNA replication a little later in the chapter. So just to remind you about genes and chromosomes, so if we're talking about eukaryotic organisms like us, we have multiple linear chromosomes, and our genes are arranged in a linear fashion on this DNA. If we're talking about prokaryotes, which we will talk about, bacteria, they just usually have one circular piece of DNA. And we'll talk about bacteria when we talk about, replication, because those were the model systems that were used first. And so you kind of have to understand what the genetic material kind of looks like, how it's organized in both eukaryotes and prokaryotes. Okay. Just to give you an idea of the amount of DNA that you have, humans, we have 23 pairs of chromosomes. Our genome is about 6 billion base pairs in size, and we have somewhere between 20 and 25,000 functional genes. So we have a pretty large genome. Okay. So in the 1860s, it wasn't known yet what the molecule of heredity is. And so the big question was, is it DNA or is it protein? And again, scientists were not sure. Protein is maybe a more complex structure. When we think about proteins that are made of amino acids, there's 20 different amino acids. So it's a large amount of complexity that proteins can have. DNA, on the other hand, is made up of 4 bases, so maybe a more simple molecule. And so scientists were kind of leaning towards proteins until studies were done that provided evidence that DNA is the molecule of heredity. So let me kind of walk you through this history. So DNA was first discovered in the 1860s by a scientist named Friedrich Miescher, and his story's pretty interesting. He would take bandages from individuals who had pus-filled wounds. So he collected pus-filled bandages, and he would scrape the pus off. And so in that pus were leukocytes. And so that word is right here. Leukocytes are white blood cells, right? So an immune response was happening. Those people probably had an infection. He would take those leukocytes and he would crack them open and he would isolate this stuff that would precipitate out. And he called that material nuclein. And he didn't call it DNA at the time. He called it nuclein. It was DNA. So he gets credit for being the first person to isolate DNA. And then in the early 1900s, we've already heard of Thomas Hunt Morgan's work out of his lab. He was able to figure out that genes are arranged in a linear fashion on chromosomes. And again, he showed that chromosomes, or we knew that chromosomes were made of DNA and protein, but the question still remained, okay, so now we have this substance and we know that it's made of DNA and protein. Which one is going to be the molecule of heredity? Okay, so here's the first experiment. This was done by Griffith in the 1920s, so 1928. What he showed in his experiment that I'm going to show you on the next slide is that genetic information, whatever it is, this molecule of heredity, whether it's DNA or protein, it could be transferred from one bacterial cell to another horizontally, so rather than by descent. So he showed that bacteria can actually pass genetic information between each other, and bacteria could also pick up genetic information from the environment. He called this transformation, okay, and this was not seen before Griffith's experiments. He used, his model organism was a bacteria called Streptococcus pneumoniae, okay, and so Streptococcus pneumoniae, he's going to use mouse as his model system, and he's going to infect the mice with Streptococcus pneumoniae, and if it's the pathogenic strain, the mice are going to die. Okay, so he's going to use the bacteria Streptococcus pneumoniae. There are actually 2 strains of this bacteria. One strain is called rough, represented by R, and the rough strain is not pathogenic. It doesn't cause disease. The other strain, which is called smooth, which we represent with a capital S, is pathogenic. So it does cause disease and death in his mouse model. So here's the experiment. Okay, and again, we call these transformation experiments. And so let me walk you through this. So starting all the way over here on the left, if Griffith took pathogenic Streptococcus pneumoniae, so the smooth or S strain, and he injected those into mice, and he comes back a few days later, he's going to find that the mouse has died. He has succumbed to those pathogenic bacteria, has disease and died. That's what we expect. Then he had a separate experiment where he took non-pathogenic bacteria, they were living, and he injected those into mice, and he came back a few days later and nothing's wrong with the mouse. And so those two results are completely expected based on what we know about Streptococcus pneumoniae. Okay, he then took the pathogenic S strain of Streptococcus pneumoniae, and he applied heat to them. So he put them into a test tube and lit a flame under the test tube, and we say he heat killed them. So these are dead Streptococcus pneumoniae. Okay, he injects those into mice, and as you would expect, the mouse lives, right? Because if you have pathogenic bacteria that you kill, they can no longer cause infection. So the mouse lives. That makes sense. Here is the last part, and it's the most important part. So he took those heat-killed pathogenic bacteria that did not cause disease on their own, and he incubated them with the non-pathogenic bacteria, which again, did not cause disease on their own. He incubates these, and then he takes that incubated mixture and he injects it into the mouse, and the mouse dies. Okay, he then takes the mouse and extracts some of the bacteria, and he finds that what he gets are pathogenic bacteria out of this mouse. And so he says, how could that be, right? So again, this is called transformation. And so what happened here is the non-pathogenic bacteria picked up DNA from the pathogenic bacteria that were dead. They picked it up and they expressed it. And so they were transformed. And so that means that they had a change in genotype. They picked up foreign DNA. And they had a change in phenotype. They were now pathogenic themselves. Okay, so again, no one had showed this before Griffith. Did he show that it's DNA itself? No, he failed to carry out the experiment far enough. However, in 1944, a few years later, another lab, and the three scientists that get credit here are Avery, McLeod, and McCarty. They took Griffith's experiment a step further. And what they did is they took the pathogenic bacteria from the dead mice, they had many, and they took and they cracked open the pathogenic bacteria, and they isolated and separated out the protein and the nucleic acid portion. And so now they had just protein and they had just DNA. Okay, and so again, the question that they're asking is, what is it? What fraction? Is it the DNA or is it the protein that causes transformation? And so they redid the experiment, right? They took non-pathogenic bacteria and they incubated them with just the protein fraction. They injected that into the mice and the mice lived. So it's not the protein. And then they took non-pathogenic bacteria again and incubated them with just the nucleic acid portion from the pathogenic bacteria. They injected that into mice and the mouse died. And so again, this was really the first evidence that it was the DNA, the nucleic acid, that caused the transformation and that caused death here in this case. And so it must be the nucleic acid fraction, the DNA, that is the molecule of heredity. So there was good evidence. The scientific community was not convinced. And so in 1952, right around the time where we were figuring out the structure of DNA, another lab did some similar experiments, but using a different model system. And so the two scientists involved here were Alfred Hershey and Martha Chase. And again, this was in 1952. Same idea, different system. So here's their model. They used bacteria. So in this picture over here, this yellowy structure, this is the plasma membrane of the bacteria, and this would be the cytoplasm of the bacteria. So they had bacteria, and then they had what are called bacteriophages. So if you see these things that look kind of like aliens, they're green, right? There's this head region, this mid region, and then down here, it's their attachment region. Okay, this is a bacteriophage. What the heck is that? This is a virus that infects bacteria. Okay, even bacteria get infected by viruses. No one can escape viruses. Bacteriophages are very simple. Okay, they are going to have DNA inside, and that DNA will be surrounded by a protein coat. Okay, and in this picture, you can actually see, so here is the bacteriophage, right, sitting on top of this bacterial cell, and you can actually see this green material. It's coming out of the bacteriophage and going into the bacteria, here, here, and here. That's actually the DNA of the bacteriophage, which is pretty neat. Okay, so we know now that bacteriophages, these viruses, inject their DNA into the host cell. And then once that DNA is inside the host cell, it's able to replicate and make more virus. Okay? And so viruses, even the viruses of bacteria, they can't replicate on their own. They require a host cell. In this case, the host cell is a bacteria. Okay, so understanding the model here is important. So this is their experiment. And again, this is more evidence that it's DNA and not protein that is the molecule of heredity. So let's start over on the left. We really have two separate experiments. So on the top, we have one experiment, and on the bottom, we have kind of a separate experiment. They're related, but they're different. So on the top, what this scientist did is they took bacteriophage, right, and grew them up with host cells in media that contained radioactive phosphorus. So here, this P32, this is radioactive phosphorus. And so why would they do this? So they're trying to radioactively label all of the DNA. So DNA in its backbone, we're going to see in just a little while, that DNA is made up of a sugar phosphate backbone, so lots of phosphate groups. And so radioactive phosphorus will be incorporated into all of the DNA in this organism, or in this virus, I should say, not organism. What doesn't get labeled is the protein. Nothing else gets labeled, only the DNA. Okay, so that's the top experiment. I'll come back to that in just a second. In the bottom experiment, they did the reverse. So what they did is they incubated, again, the virus, allowed it to replicate in the presence of radioactive sulfur, so S35. And so there's really not a lot of sulfur, or any, in our DNA. There's no sulfur in our DNA. I should reword that. But there's a lot of sulfur in proteins. And so in this second batch, the scientists were radioactively labeling protein. So in the top, we're radioactively labeling DNA. And in the bottom, we're radioactively labeling protein. Okay? That's how we start. So let's look at the top. So what they did next is they took bacteria and they incubated the bacteria with the virus that had its DNA labeled. And they incubated it long enough for infection to occur. They then took the bacteria and actually put it in a blender to shake off anything on the surface. So any viral particle that wasn't in the cell yet would fall off. Okay? And they did the same thing down here, except for, again, remember, this was virus labeled with S35, so proteins were labeled. Top, we're labeling DNA. Bottom, we're labeling proteins. So in this top one, after they sent this through the blender, they put it into a test tube, and they spun it in a machine called a centrifuge, which spins samples at a high speed, and it separates samples based on density. And so the heavier bacterial cells will go to the bottom, and the lighter bacteriophages will go to the top. they then removed this top layer. Okay, they took what was in the bottom here, these bacteria that have radioactively labeled DNA in this case, and they plated these and created a culture, and then they looked at the resulting viruses that were made. And in this top example, they found radioactivity, right? And so what that told them is in the very beginning here, it was the DNA that made it in, and it's the DNA that allowed it to replicate. In the bottom, same thing happened, right? They incubated their radioactively labeled viruses, their proteins were radioactively labeled, with bacteria, allowed infection to happen, shook off anything on the surface using a blender, put it into a test tube, spun it to separate out these pieces, took the bacteria here at the bottom, plated them, and they found no radioactivity. And so again, this was the second experiment done that provided really good evidence that it's DNA that's the molecule of heredity and not protein. And now we know today that that's very true. Okay, I'm going to leave you with this very last slide. Again, a little bit more history kind of leading up to the structure of DNA, which we'll talk about in the next part of the chapter. I want to tell you about one more scientist that did work right around the same time. So in the 1940s, there was a scientist named Erwin Chargaff, and he was looking at DNA content from a variety of different species. And his findings were really important and used when determining the structure of DNA. And so what he's found, again, he's looking at DNA from a lot of different species. He found that the amount of the 4 nucleotides found in DNA, and we'll talk about these more, they are adenine, thymine, guanine, and cytosine. So there's 4 bases, 4 bases of nucleic acid. These 4 bases were not found in equal quantities. Instead, in every species he looked at, he found that the amount of adenine was always equal to the amount of thymine, and the amount of cytosine was always equal to the amount of guanine. And so he came up with what we now know as Chargaff's rule. He said that in this molecule, it must be that adenine is somehow associated with thymine, and cytosine is somehow associated with guanine. And so now we call this Chargaff's rule of base pairing. And so when we get into structure, we'll talk about this more, but I want you to remember this. Adenine always pairs with thymine, and cytosine always pairs with guanine. And I remember this mnemonic, A to T2, C to G3, okay? And again, I'll be talking about that in the next part of the chapter when we look at structure. That's it for this part.